everyone. Today we are going to learn how to make zebrafish models for studying various diseases. Let's learn about pathogenesis first of all. So pathogenesis is the study of the disease and how the disease develops in the body. And this requires animal models for several reasons. First is for human virus diseases, another host is required for the experimentation. Next reason is in vitro or in silico systems cannot simulate the complex response of a living host with its innate and adaptive immune response. So that is why we need something which is an in vivo system. In vivo system is when we study diseases inside the body of an organism. In vitro is when we study the disease outside the body of an organism, and in silico system is completely over computers. So only an in vivo system can reproduce the sometimes unpredictable responses of a natural host. Otherwise, it is very difficult to make it out in vitro and in silico system. So pathogenesis studies in animal models are also of great importance to applied science, since they constitute an essential foundation for the development, assessment, and production of drugs and vaccines to treat and prevent important human and animal viral diseases. So while choosing an animal model, we have to take care of multiple things, and it is a very complex decision since it involves scientific and practical consideration. So in many examples, it may be best to use several animal models to address different aspects of pathogenesis or to test candidate treatments or preventive interventions. The mouse has become the default animal for many virus infections because there is a vast scientific database and a large set of reagents, and because it is the least expensive animal model as well. So, however, there are many situations where other animal models are required to best address the experimental questions. The use of animals in research is required for the production of new drugs and vaccines and plays a critical role at several stages in the developmental process. Small animals may be used to screen large number of candidate drugs or immunogens for potential efficacy and also for unwanted toxicity. So let me give you an example here. Let's say I want to mimic a disease model wherein I want to make a model for, let's say, throat infection. So we all know that we cannot do that on rat models, right? For that we would need an animal model which is closest to the human species, at least more closest than the rat model, right? So for that, animal or the other chimps, orangutans, they may be useful. But if I want to do something in liver or lung, if I'm working on a disease which is related to this, or to brain or to intestine, the other diseases other than the throat infection, then the rat model will be very good. So larger species, particularly non-human primates, they may provide models that better simulate human disease, where promising products can be tested to select those qualified for human trials. In special instances, and the animal rule may be applied where data from two animal species substitute for human trials. So now animal-based research is entering a new and exciting phase because of a variety of evolving methods to select mice with specific and variable genetic backgrounds or to manipulate the mouse genome. So now it is called it. So now in the today's time it is possible to delete or inactivate specific genes, and when we make these type of models, these are called as knockout models. Or otherwise, we can insert few genes in the genome of the rat or the other animal model wherein we are working, and this type of model is called as knock-in model, or to introduce specific mutations using an armamentarium of techniques. So these methods enable the assessment of the individual host genes in response to the viral infection. So when we knock out or knock in any specific gene, we are trying to understand how the gene is playing role in a certain viral infection or in a certain disease condition which is not from the viral background. So recent developments in omics and systems biology have made it possible to record a vast number of discrete host responses during a single infection, enormously enhancing the dissection of a very complex process. So another technical advancement is the new set of imaging methods to visualize both viral invader and host responses in the living organism, or to capture three-dimensional images of intact functioning tissues. Together, these new technologies are opening an expanded vista of virus-host interactions, which will take viral pathogenesis studies to an increasing level of sophistication. Now let's understand how to make models in zebrafish, because now we are also using zebrafish to model many of the diseases which comes from humans. Let's say Alzheimer's, Parkinson's, liver cancer. These are all the diseases which people have been modeling. People also have been, testing the regenerative capabilities of zebrafish. And diabetes is another disease which people study in zebrafish. So now let's see how to make models in zebrafish. So first method is the oral method, wherein we give toxin directly in the water and we keep our subjects in the water for a specified period of time. So this type of model is one of the type in oral section. So this is an oral model. Another way in which we can do oral modeling is this by the use of an oral gavage. So now let's understand what is the difference between two of them. Whenever you are trying to model a disease or a project, when you're working in a project wherein you are trying to see or assess the the impact of certain metal lines or something which comes from the environmental background on the health of zebrafish. So you can directly put the medicine or the toxin in the water and you can let it work, because this is how the the toxin is going to reach to the environment. But if you are working on understanding of how the toxin is going to behave in the body of the fish, this is one of the method which one can use, which is to use the oral gavage system. So for that, you can see the image on the right hand side of your screen. You have to hold your fish first of all in some type of system, and the system which you can use here is you can use a foam and you can put your fish in there, and then you can set up the medicine in the specific system which are designed for the zebrafishes, or otherwise local made if you can, and if you have the micropipettes with you, and keep the dose ready, and you can directly inject it in the mouth of the zebrafish. But then that will require a skill to do that, otherwise you will end up damaging the organs of the zebrafishes. So now the other method to make models in zebrafish is either via intramuscular injection or by intraperitoneal injection. So intramuscular injection is when we give the injection in the muscle area of the of the fish, and the intraperitoneal is when we inject the toxin from the interperitoneal cavity of the subject. So you can see you can see in this image that the injection is entering into the peritoneal cavity of the fish, and this is the place wherein the compound will be injected in the body of the fish. This excellent picture has been taken from JOVE, which stands for Journal of Visual Experimentation. It is a very good journal, and there you can see you can visually see the experiments being performed. So now you can see how intraperitoneal injections work. This is the place where the injection has to be loaded. And in our lab, what we do is, since we have to hold the fish in order to do this, so we make use of foam again, and otherwise you can do it through other means also. So this is a local made means, which is you can have a cotton cloth with you, dip the cloth in the water, and then you put the cloth around your fish. So fish finds it difficult to remove the cloth from the body and stays still for that period of time. So now you can hold your fish and then you can put in the toxin through the interperitoneal cavity, and this is the another way in which it can be done. So this is the image. Now this is the place wherein all the organs of the zebrafish are located. So whenever the injection is being done, and now we are proceeding for dissection of the fish after a few days, whichever you have planned. So this is the area wherein we have to focus. So these are the step number 1, 2, and 3. So you have to put a cut through scissor, and I'll take my mouse here. So you'll have to cut the interperitoneal cavity very lightly. Do not damage the organs of the fish. Then you cut in this manner and then go up in the second stage, and then come back cutting the skin of the zebrafish in this manner. And when you will do it, it will open like this. And if you will do this dissection on a dissection microscope, you will clearly see the swim bladder, the testis or the eggs, depends on the sex of the zebrafish, intestines, liver, heart. And when you will remove all these organs, then you will find the kidneys, which are basically lined up on the over the the backbone of the fish. So this is the place where you will find all the organs, and this is how you do the dissections. So I hope this video was useful for you. Thank you for watching.